Hello, Mr. Allman here, and this is your Allmancast on proteins. As we've covered previously, proteins are a polymer made up of amino acids. Their structure is varied, and proteins are extremely versatile. They're easily the most important biological molecule to all of life, because they're the ones that do all the work of processing energy, transporting, communicating, and in essence, keeping us and all other living things alive. The amino acids are joined together by what's known as a peptide bond. The condensation reaction is the process by which an oxygen and hydrogen from the carboxyl end of one amino acid and a hydrogen from the amine group of a second amino acid are going to be removed, forming water as a byproduct, with a new covalent bond called a peptide bond being formed to join them together. There are 20 amino acids that are encoded by our DNA. Meaning that a particular sequence of DNA nucleotides is going to correspond to one of those 20 amino acids. There are many more amino acids, there are actually hundreds of known amino acids, but these are the 20 that we're going to focus on because we see that information in DNA is used to be able to build them. As we look at the structure of those different types, the two first important groups are those that have a positive charge. These are also known as the basic amino acids, and two that have a negative charge. These are going to be the acidic amino acids. Those that are polar but have no charge, so these are obviously going to be hydrophilic because they contain a large amount of nitrogen and or oxygen, which are atoms that are going to have polar bonds, and thus these are going to interact. They would dissolve in water if they were on their own, and these are going to be located in parts of the protein that are exposed to water. Cysteine is a special case because it is polar, however it has sulfur in it, and so cysteine is going to be important in informing the tertiary structure and the quaternary structure of amino acids, and we'll come back to that in a few minutes. Glycine and proline are also special cases. They're going to tend towards the non-polar side, however. And then we have a large number of amino acids with hydrophobic side chains. So hydrophobic side chains are going to come about because of the predominance of hydrocarbons. These non-polar molecules in the side chains is going to make the molecule as a whole repel water, and so you're going to find these in places such as the interior of the plasma membrane or the center of the protein, where water is not going to be able to come in contact with them. One thing that you'll notice as you look at this chart and similar ones is that we have several different ways of being able to represent amino acid names. We have the full name, alanine, isoleucine, leucine, etc., and we have a three-letter abbreviation, which is going to be have some shortened form of the name, and then finally a single letter abbreviation, which sometimes will correspond with the beginning of the name. Because there are several amino acids that start with the same letter, you might see that that particular letter, in the case of tryptophan, the W, doesn't necessarily correspond to what it begins with. That single letter is often just used as a shorthand because it's a little bit more convenient if you have a long list of amino acids to use just a single letter to abbreviate it with. All proteins have a directionality depending on which end of the chain you are looking at. So as those peptide bonds join together these new amino acids, you can see on the bottom here we have a tripeptide, meaning we have three sets of amino acids. This is the N-terminus end because the part that is exposed is the amine group, which contains nitrogen. On this side, this is the C-terminus end because we have the carboxyl group with the carbon exposed on the opposite end. So the N-terminus on the left and the C-terminus on the right. You can see the side chains here in yellow and in green. Those are a part of the chain, but this N and C terminus, it doesn't have to do with the side chain, it has to do with what's called the backbone of the polypeptide. Now, protein structure is broken up into four parts. What I'm going to describe here is going to be more relevant to HL bio students, so you're going to want to make sure to pay attention and take some notes as I move through these four levels of protein structure. Summarized, you can see the image here on the left. We have the amino acid sequence of the primary structure. we have the secondary structure, which are these regular repeating patterns of alpha helix and a beta sheet. We have the tertiary structure, which is going to be the folding of those secondary structures back on themselves. And then a quaternary structure is going to be the combining of multiple subunits of a particular, to assemble a particular protein out of multiple parts. To look at the primary structure, I'll use the example of human insulin here. This is going to be the sequence of amino acids. So this is still just a long unfolded chain of amino acids to make up what's called a polypeptide. It's not really a protein yet because without its particular three-dimensional shape, it doesn't really have any function yet. So that's why we just call it a polypeptide. The polypeptide, you can see each one of these circles represents an amino acid. So by counting up the number of circles, you can get the total number of amino acids. In this case, we have 30 for this purple polypeptide, and we have 21 in this blue polypeptide. The cell knows the order of amino acids because this is information that's encoded within DNA. And in a future unit, we're going to cover exactly what the mechanism is by which a ribosome will be able to synthesize one of these polypeptide chains based on the order of bases in the DNA code. A secondary structure is going to be when some basic folding or basic coiling begins to happen. And this is going to be based on the hydrogen bonding that's going to occur between atoms in the backbone. Particularly as you can see in the image here, you can see that there are these dotted lines. The dotted line represents the hydrogen bonding that occurs between this red atom here, which is the oxygen, part of the carboxyl group, and then you have the light blue one. This is going to be a hydrogen, which is attached to the dark blue, which is the nitrogen. So essentially you have hydrogen bonding happening between the carboxyl group of one end of one amino acid and then the hydrogen on an amine group in another amino acid. And so as those multiple hydrogen bonds form, it causes the amino acids to be able to twist back on themselves, and that creates this coiled pattern, and that purple ribbon represents the shape, which is called an alpha helix. So a helix structure is something that we commonly see. There's also going to be hydrogen bonding if these things don't coil, but if they run back and forth against each other in what are called beta sheets. And so you have one set of amino acids on the bottom, a second set of amino acids above that, and hydrogen bonds hold those amino acids in place. It's called beta pleated sheet. It's a sheet because it's flat, and it's pleated because it does have a little bit of a wave up and down. If you imagine if you folded a piece of paper, those creases are where the amino acids are kind of going up and down a little bit like a wave. So these side chains are not involved with this, which is why the secondary structure is not dependent upon the type of amino acids. It is going to form based on the parts of the amino acid that are in common between all proteins. The tertiary structure, this is where a protein really is beginning to form and it begins to get its function, because the tertiary structure is the three-dimensional shape of that particular chain. It's also known as its conformation, based upon the interactions of the side chains. So now this is not the backbone, but this is all of those special R groups, whether they are polar or nonpolar, charged or uncharged. It's the ways in which they are going to interact with each other. A few examples of that you can see in the image up here. You can see that there is a sulfur-sulfur bond. This is called a disulfide bridge formed between two cysteine amino acids. Those cysteine amino acids could be far apart from each other on the polypeptide chain. So they're far apart based on their primary structure, but once the thing is folded up, they end up becoming very close in contact. You have hydrogen bonding here, so this is going to be hydrophilic or polar amino acids interacting with each other. These are hydrophobic amino acids, and so they are going to also interact with each other because they're going to be repelled by the water that surrounds the protein. And then finally you have an acidic and basic amino acid based on those opposite charges being attracted towards each other and neutralizing each other. You can see in this image on the left, this is the insulin protein again, and you can see each of those coils represents an alpha helix. You can see that usually an arrow, which I'll zoom in here, represents a beta sheet. And so that combination of helices and arrows, the thin little spaghetti-like part, is a part of the amino acid chain that's not a helix or a sheet, but often connects, maybe just a sequence that connects those helices towards each other. The tertiary structure is going to be the ways in which those helices and sheets all fold back on themselves. So you have a couple different ways of representing this. On the left, this shows a space-filling model that shows colored circles to represent all of the atoms involved, so you can get a sense of the three-dimensional structure, whereas the one on the right is going to highlight all the secondary structures that make up this complex three-dimensional shape. So important interactions, again, we have some covalent bonds like the disulfide bridge. We have hydrophobic interactions, we have hydrogen bonding, and then we also have ionic bonds from the attraction of those opposite charges, all working together to make the overall tertiary structure. Finally, quaternary structure. This is not something that's found in every protein, but many proteins have one. This is the specific three-dimensional shape that comes about as multiple polypeptide chains join together. Insulin, in its functional unit, is a hexadimer. Hexa referring to 6, and dimer referring to that we have pairs of identical parts coming together to form this 6-part protein. So the insulin hexadimer, you see each color represents one of the 6 polypeptide chains that makes it up. Within each polypeptide chain, if we look at the yellow example here, you can see that there is a helix, then there's going to be a sheet, and those will turn back on themselves. So the quaternary structure is often going to be necessary to form in order to actually develop an active site or to develop the actual three-dimensional shape that's needed for that particular protein to accomplish its function. By themselves, those single polypeptide chains wouldn't necessarily be able to do anything, but it's the way that they form together that ultimately makes them functional. Protein function, to go back into some standard level content here, protein function is varied. And so again, most everything that happens within the cell requires proteins to be able to do it. We have storage proteins to be able to store amino acid, like casein in milk is going to be a source of protein which has a purpose of food. We have hormones such as insulin, which I just mentioned. We have structural proteins, so your hair, the collagen that's in your skin, a lot of the proteins that are going to be found in connecting the cytoskeleton to the plasma membrane are providing cellular or tissue level structure. Defensive proteins such as your immunoglobulins, or aka antibodies, are going to help to recognize pathogens and help your immune system to destroy anything that's trying to make you sick. We have transport proteins like hemoglobin or things like glucose transporters. We have contractile proteins that are involved in movement. So we have actin and myosin, which are 2 proteins that work together to be able to provide muscle movement. We have receptor proteins like those, what's shown here is a signaling molecule being released by a neuron, so that those are could be neurotransmitters that are proteins that are released. And then you have enzymes, and enzymes, things like lactase or amylase, that are performing either a catabolic or an anabolic function to be able to catalyze chemical reactions. And one example of a little bit of a preview into something that we will cover again in a future unit, and that's going to be the protein hemoglobin and how the primary structure ends up becoming very important in a common disease called sickle cell anemia. So hemoglobin is really important. Hemoglobin does have a quaternary structure. There are 2 alpha subunits represented in dark blue, and then you have 2 beta subunits represented in green. Each of those holds one heme molecule, which has an iron ion embedded in it. It's that heme group that's going to attract oxygen, So hemoglobin is a transport protein that's going to attract oxygen and carry that in your red blood cells. So this is going to be a quaternary structure made up of 4 polypeptide chains combined together. Now, for people who have the sickle cell trait or sickle cell anemia, their DNA has had a mutation that has placed a hydrophobic amino acid in the place where a hydrophilic amino acid used to be. So the hydrophilic amino acids, which are usually located on the outside of the protein, would interact with the water that's going to be in the rest of the cell. But when that's changed to hydrophobic, now there is a repulsion between the water-based environment and that protein. As a result, the proteins are going to stick together. And so you can see that this fiber is produced because that one hydrophobic amino acid on the surface of the protein is going to cause all those proteins to stick to one another. And once they stick to each other, we lose that nice round shape that we expect in a red blood cell, and now we have this curved shape that's going to, this sickle cell shape that's going to cause a lot of physiological problems in the person who has that. So again, it's one change, so you can see the glutamic acid, a hydrophilic amino acid, being changed to valine, which is a hydrophobic amino acid. That ultimately changes the tertiary and quaternary structure of the protein, because that folding of the protein is based upon the side chains of the amino acids that it's made up of. That's obviously going to cause some system-wide problem, particularly in circulation, because of inability to carry oxygen, because of the change in shape of the protein and the damage to blood vessels, and issues with joint pain that's going to cause. A final note here on what can happen to proteins and their tertiary structure. Denaturation is going to be any change in the structure of the protein. As that structure changes, a small change might reduce the function of the protein, or a large change in the structure is going to cause the protein to completely lose its ability to do its job. So denaturation is going to ultimately harm the ability of the protein or prevent the protein from doing what it's supposed to do. In some cases, proteins will renature, but they may renature into a different shape. And if they have a different shape, that often is going to mean that their function is still going to be lost. Finally, looking at visualizing proteins, so you can see a few different things here that you've seen thus far in some of the previous images that I've shown you, with the most common two being the ones that you'll see here. So again, the arrows, sometimes you just see kind of a flat ribbon. Those ribbons are going to be the beta sheets. When you see the curved structure, the spiral is going to be an alpha helix. Those are both examples of secondary structure. And then you have the thin noodle-like part, which is going to be some sequence of the protein that doesn't have one of those two shapes. This doesn't help you to see the three-dimensional shape as much as the one on the right does. So the one on the right shows you now with all of those atoms actually drawn in, you can see that this isn't like a porous thing, but actually atoms fill up all of that space in the center. And so the part that's on the outside of the protein, that's going to be where the active site, if it's an enzyme, is going to be located, like the example here that you see with this groove. That's the place where on this enzyme a substrate will be able to fit in. But that takes us into enzymes, which is the subject for a different video. Thank you for watching, and please let me know if you have any questions.